I am Dhinal Patel, Assistant Professor, SMC College of Dairy Science, Anand. I am the content writer of paper number 13, Food Additives. In module number 26, Enzyme Applications in Fruits and Vegetables, Brewing and Wine Processing. As we know, the enzymes are very helpful in a different fruits and vegetable processing as well as the beer making and obviously in a wine processing industry. So let's see them one by one. Introduction. Numerous fruits and vegetable juices came into the market. In a fruit, pectin is the major component to break down in order to facilitate the juice extraction. The vegetables have a high content of dietary lignocellulosic fibers which hinders the juice extraction. The vegetable juice processing therefore requires more celluloses in addition to pectinase to reduce the viscosity. Rapidase is recommended for vegetable juice extraction as it contains pectinase and celluloses. The juice's carotene content is not decreased by the enzymes and the cloud is stable for the duration of the juice shelf life. Now looking into the advantages caused by the enzyme applied to the fruit and vegetable processing. The enzyme increases the overall productivity. The juice which is extracted from the fruits and vegetable remains stable for a long term without additives. It lowers the risk of microbial spoilage, reduces the oxidation and improves the juice and concentrate shelf life. It also lowers the energy consumption, that is electricity, steam as well as the water. It reduces the waste flow by maximizing the fruit use. It also reduces the dependency on chemicals used in equipment cleaning as well as the product. Now looking into the new developments for a citrus peeling, the pectinases are used to soften the peel. The current industrial practice is starting with a pectinase treatment of a whole fruit that is to be followed by a vacuum infusion treatment with a pectinase solutions like rapidase intense and Lysozyme containing a pectin stearase and polygalacturonase. Thereafter, the peel can be removed easily within 1 to 2 hours and the enzyme solution can be recycled. As far as the whole fruits application concerned, the most pectinases preparations consist of multiple enzymes leading to a weakening of the pectin polymers. For example, dimethylation by pectin methyl esterase, that is generally known as a PME, which is to be exposed the homogalacturon backbone, which will be further degraded by the enzyme like pologalacturonase and rhamnogalacturonase, causing a physical weakening of fruits. The firm fruit concept is based on a use of PG and RG free PME in a combination with a calcium which binds to the freed pectin acid in situ to form insoluble calcium pectase. Looking into the immobilized enzymes, the traditionally enzymes in a fruits and vegetable processing industry are applied as a liquids or a powder, while enzymes in a pharma are often immobilized. Although immobilization add an additional step and thus cost in the preparation of the enzyme. That improved characteristics like lower pH optimum and increase half time through recycle can turn this into an attractive opportunity. Next, the next is preventing the haze formation. The addition of gallic acid in combination with various proteases also reduce the haze formation drastically but retain much more of the beneficial phenolics. The best performing enzyme in a enzyco fungal acid proteases produced from the Aspergillus niger, which reduce only 12% of the phenolics, while the traditional method reduces up to the 30% of the phenolics from the subsequent juices. The next application includes preventing a cloud loss. To produce a cloudy juice, the process from extraction to pasteurization must be fast to minimize the effect of plant indigenous enzyme like pectinases and oxidases. The alpha amylases and the glucoamylases can be added to reduce the starch content as dissolved starch retrograde during cold storage and can form a precipitates. After seeing the application of enzymes in a fruit and a vegetable industry, now let's move forward to a brewing industry that is belong to a beer production. As we know, enzyme is very useful in a different stage of beer formation, maybe it from maturation, from mashing, from lautering. Even though using enzyme we can produce a low calorie beer also. So let's see its application. The next is enzyme application for a brewing. In a brewing, now we are going to discuss about the application of enzyme in a malting. During the malting process, the enzymes native to the barley, that is amylases, glucanases, proteases, and hemicellulases, are activated. As a result, the malted barley is made from amenable to milling and starch carbohydrate extraction. Apart from the endogenous enzymes present in a malt and a barley, no exogenous enzyme are applied in a malting process. Next application in a mashing, the role of enzymes in a mashing. During the malting process, some enzymes have already been active in a degrading the various polymeric substrates. The exogenous, that is added enzymes such as proteases, amylases, and glucanases can help to increase the degradation of polymeric substrates. The most important benefit of using exogenous enzymes is that they give the brewer the required tolerance in a using his malt. Protein converting enzymes. Mashing usually starts with the degradation of malt protein by means of proteases and peptidases. These enzymes are referred to respectively as exo and endoproteases and that produce respectively a free amino acid and a peptides and a smaller proteins. This process step is also called as the proteolytic stand. Looking into the cell wall degrading enzymes, the degradation of cell walls is carried out by glucanases. Incomplete cell wall degradation have the negative impact on mass filtration, that is generally known as a lautering, beer filtration process steps, colloidal stability of the final beer, that is generally we can call it as a haze formation. The most important polysaccharide to degrade is a beta glucan. The xylene is usually already sufficient to degrade in a malt. Next is a starch converting enzymes that is carried out by a range of amylophytic enzymes in a malt, that is alpha amylase, beta amylase, and limit dextrinase. From a microbial origin, the enzymes polylinases and amyloglucosidase can be added. The alpha amylase splits the polymer in an endo manner and therefore leaves a considerable amount of branch maltodextrins from amylopectin, while beta amylase is a less stable than the alpha amylase and limit the dextrinase in even a less stable form. The next is enzyme in lautering, or we can say filtration, mass filtration. The lautering and other form of mass filtration are usually performed at so-called massing of temperature of 75 to 78 degrees centigrade. At this temperature, almost all malt enzymes are already inactivated. Only a small amount of residual alpha amylase activity will be present where a relatively low mass filtration temperature is used. The amyloidic enzyme only show a limited amount of activity at this stage during the massing. Enzyme application in fermentation. The exogenous enzyme are used in a fermentation. In case of beer filtration problems, a beta glucanase can be added to the fermenter or during the maturation, that degrading the residual glucans which would otherwise cause filters to the block. The another area of enzyme application during the fermentation is to reduce the haze problem in a final beer. After analyzing the type of haze material, a fungal alpha amylase and a beta glucanase for respectively starch or a glucan haze can be applied. Now looking into the low calorie beer production, the low calorie beer can be produced with the application of exogenous amyloglucosidase during fermentation, which will degrade the dextrin to fermentable sugars. In this way, a normal alcohol or a low dextrin beer can be made from a wort containing a reduced amount of extract. The application of exogenous amyloglucosidase is combined with the fermentation of low extract containing wort or with removal of alcohol, for example, by a vacuum distillation. Now let's see the application of enzyme in a maturation. The enzymes to correct a problem, that is for filtration and a beer haze. The exogenous enzyme can correct incomplete degradation of starches and glucans, causing beer filtration problem or a haze problem in a final beer. The enhanced maturation with acetolactic decarboxylase, that is generally known as ALDC. One of the aim of maturation is to reabsorb diacetyl, which is considered a buttery off flavor in a lager beer. The alpha acetolactate decarboxylase, a microbial enzyme that is from Bacillus, converts alpha acetolactate into acetone before it can be converted into diacetyl. Next is the chill proofing enzyme. Beer will lose its clarity quite quickly. Apart from the earlier mentioned glucan hazes, that is a permanent haze, so called a chill haze, can play a major role in a beer quality. The chill haze particle usually develop through a complex of protein and polyphenols, that is also called a tannins. The enzymatic approach is to degrade the haze forming protein so the protein polyphenol complexes will not be able to aggregate over a time and the beer thus remain clear. For this purpose, the enzyme papain is typically used for a chill proofing. That's why it is also called a chilled proofing enzyme. The next application of enzyme in a wine processing. During wine processing, enzyme for pressing and maceration. The enzymes act on the polysaccharides of the grape cell wall, such as arabinins, galactans, arabinogalactans, and arabinoxylans, make it possible to optimize the usage of enzyme in a winemaking. The mechanism of action of pectolytic enzyme preparations, usually called a pectinases, as far as is currently known, the enzyme activities involved in hydrolyzing pectic substances are pectin esterase, polygalacturonase, pectin lyase, raminogalacturonases, raminogalacturonase acetylesterase, arabinase, and galactanases. The enzyme preparation for aroma liberation. The enzyme preparations containing glycosidase activity were developed to improve the aromatic profile of certain wines. These enzyme preparations are added at the end of the alcoholic fermentation and during the retapping of wines. The aroma development has to be controlled by tasting, and the enzyme action is finally inhibited by addition of bentonite. The small amount of this compound, that is 20 gram per 100 liters, are usually sufficient to block all the enzyme activity. The next application of enzyme for color extraction. The extraction of phenolic compounds generally occurs with the maceration of mass during alcoholic fermentation, and it depends on the variety and the quality of the grapes and on technological parameters such as crushing, maceration time, temperature, and pumping. The good results have been obtained on an industrial scale using enzymatic treatments. The improved anthocyanin extraction was obtained by enzyme addition at the beginning of the maceration. The other, the other enzymes used in a winemaking, that includes urinases, lysozyme, etc. The ureases is the enzyme is produced by lactobacillus fermentum. It is catalyzes the hydrolysis of urea to carbon dioxide and ammonia. This limits the formation of ethyl carbamate. This molecule, suspected to be a carcinogenic and a mutagenic at high dose rates, is predominantly urea. The next is lysozyme. Microbial stabilization in a wine is guaranteed by lowering the sulfur dioxide level in combination with a treatment with the lysozyme. The enzyme has the ability to break down the cell walls of lactic bacteria and its activity increase with increasing in the pH. The lysozyme treatment is recommended for blocking the malolactic fermentation in a white wines, stabilizing red wines after fermentation, treatment of problematic end stage of alcohol fermentation, preventing premature malolactic fermentation in wines. Now, looking into the new developments of enzyme application in a wine processing, the recent investigation have stimulated interest in another group of enzymes, that is oxidases, in particularly a polyphenol oxidases. Now, let's see its application, that is specifically in a cork cleaning. The enzyme removes phenolic compounds which can give an off flavor to the wines and is applied during the cleaning of the corks. This enzyme treatment significantly reduces the amount of water extractable total polyphenols in a comparison with the crude unwashed corks. The next application that includes a color stabilization. The treatment with enzymes such as lacases, anatanases, and peroxidases have been investigated for a juice or a wine stabilization. The fungal lacases has ability to react with a wide range of phenolic compounds, and lacase treatment can enhance the effect of conventional finning treatments. So, by this module, we come to know that how enzyme is very useful in a different industries like fruit and vegetable industry, for beer making, as well as for wine processing industry.